All right, I think we'll get started here. So thank you all for joining us at the IGN seminar series. Today we are welcoming Dr. Yatish Turiakhia, who's an assistant professor here at UCSD. Dr. Turiakhia got his bachelor's and master's at the India Institute of Technology. And then he came to California, where he's been working his way down the coast while pursuing further graduate training and starting his career as a PI here. He got his PhD at Stanford, then did his postdoc in the University of California, Santa Cruz. And then in 2021, he joined UCSD as an assistant professor. And since starting here, he's really developed algorithms to kind of investigate Pangenomes, which was really useful in assessing and surveilling strains and variants of SARS-CoV-2 in the population. And I believe today he's going to update us on some of his data on SARS-CoV-2 and maybe some other projects as well. So I'd like everyone to welcome Dr. Turiakhia. I'm sorry, Turiakhia.
You are the first student, so Turiakhia. That's okay, thank you. Okay. Thanks, Tim, for the wonderful introduction and for also inviting me here. I'm very excited to present today some of our ongoing, this talk is really about the vision of an ongoing research program in my lab and the exciting research progress that we are making in this direction. And as you can see from the title, it's on building ultra-large pangenomes. A lot of what I would talk about today is actually unpublished work, but we have some really promising results. So this is going to be the outline of my talk. In the first part of my talk, I'll highlight how in the midst of the challenging times of the pandemic emerged a significant silver lining, and that's the advent of the remarkable technology of genomic surveillance. And I had the unique opportunity to be a part of this groundbreaking innovation, and I really believe that this, along with some other related technologies, have greatly bolstered our readiness and capability to face and manage future pandemics and outbreaks. Of course, this is an ongoing work, and I will then discuss how this relates to an emerging area of pangenomics and why genomic surveillance of the future will necessitate the use of ultra-large pangenomes. And then I will discuss the limitations of existing pangenomic tools and data formats and how some of the recent work in our lab, particularly on PanMats and PanMans, addresses those limitations and provides unique advantages for genomic surveillance and other such applications. And finally, I will discuss strategies for building such ultra-large pangenomes and the applications to genomic surveillance and elsewhere. So our future work will entail a lot of acceleration work for the construction pipeline. So let me start with this emergence of real-time genomic surveillance. So I think this is a picture that all of us really understand, that the virus keeps mutating, right? And as it mutates, it's acquiring, or the mutations that it has acquired, which increase its fitness, tend to stay in the population and spread, but those which decrease the fitness tend to be weeded out. And as a result, what you see is that the virus gets fitter and fitter over time as it acquires these mutations, right? These mutations that tend to increase its fitness. So it's really important for us to monitor how this virus is evolving in order to take appropriate measures to deal with it and prevent its harmful effects. So as a result, genomic surveillance is really the tool which helps us monitor the mutations that the virus is acquiring. And we've been doing this for various pathogens. For example, if you look at RSV, this is how the number of genomes which have been deposited on public databases have grown over time, right? And as you can see, the rate at which we are depositing new sequences keeps on rising rapidly every year. But as far as the SARS-CoV-2 virus, or the COVID-19 causing virus was concerned, it really knocked off the park all the previous genomic surveillance work that we've done. In fact, if you look at it on the same linear scale, you see that all of the pathogens really pale in comparison. We actually got millions, in fact, more than 16 million now sequences deposited for SARS-CoV-2 in a matter of just 3 years. So this really changed the nature of genomic surveillance that we've been doing for various pathogens, and it has had it has contributed to really important insights. For example, we've been tracking exactly how the lineages of the virus have been evolving. We had earlier we had alpha, then delta, Omicron came later, and so on. And the genomic surveillance efforts have really helped us in monitoring it. But more importantly, we are also now updating our vaccines based on the way the virus has been evolving, right? So if you look at the most recent vaccine update that we have, that is using monovalent Omicron variant XBB.1.5. But if you look at the most recent surveillance data, you will see that this particular variant it barely exists in the population. So it has faded away, and other important variants have taken over. And so FDA is predicting that COVID-19 vaccines, just like influenza virus, will have to go through these frequent updates and more like an annual update that we'll have to do. And for this as well, genomic surveillance plays a very important role. So let me just introduce first the paradigm shift that we caused by some of the tools that we introduced in this particular space. And one of the contributors of this paradigm shift was this idea of online phylogenomics. So the idea here is that previously the way people used to track the evolutionary and mutational dynamics of a pathogen was they would take all the sequences and then build a phylogenetic tree and then try to see how the virus has really evolved. But the way we do it with our Usher toolkit is that let's say on day one, we had a multiple sequence alignment and a phylogenetic tree. We construct out of it using our tool, which we call Usher, a mutation annotated tree. So what that means is you take the multiple sequence alignment and you sort of infer which mutations have occurred on individual branches of this particular phylogenetic tree, right? And then the process actually gets simpler, that in the next day, you keep the phylogeny that you had on the earlier day fixed, right? So it's already mutation annotated and a fixed phylogeny. But as you get new sequences, right, which got deposited, for example, on public databases, we would take that and just place those new sequences without disrupting the existing relationships of the previous phylogeny, right? And so this way the tree keeps growing over time and helps us keep track of the emerging mutations as well, right? So that's basically the idea. So it's very different from what people used to do earlier, which is this de novo phylogenetics, which means to take all the sequences and rebuild the phylogeny as you have new sequences. Rather, what we do is we take the previous day's tree and the sequences, and we just incorporate new sequences on that existing tree. So Usher, in fact, stands for ultra-fast sample placement on existing trees. And as a result, the tree keeps growing, and this is a lot more computationally efficient strategy as compared to doing everything de novo. And in fact, people did try to do de novo phylogenetics earlier on. So for example, we had collaborators who were using IQ-tree for building SARS-CoV-2 phylogeny every day. And in fact, this pretty much saturated at around 10,000 genomes, which is when IQ-tree runtime hit 24-hour mark. What that meant was it would not be able to update as new sequences kept up and would not keep up with that data, right? So then they made some changes, which was kind of doing what we did, which is the online phylogenetics framework. But even then, after they reached around 10,000 or so sequences, they actually gave up because it was just becoming very difficult for them to keep scaling. Which is actually 100,000 is about 1% of what we see, less than 1% of what we see today. Whereas if you look at Usher, it has really kept up with the pace at which we've been able to generate this genomic surveillance data. So this is showing how many sequences were, the blue line is showing how many sequences were deposited on GISAID, and this is how many sequences we have on the Usher tree. This tree, by the way, we keep releasing every single day. And so you can see that we closely track it. The reason why a little bit of a gap exists is because of quality control measures that some of the sequences are really bad and error prone. So we actually weed them out of the tree. But more importantly, rather than just maintaining this tree, we actually also created this web interface called usher.bio. Where what you can do is we maintain this tree, but if users have some sequences which they have just collected, they can upload those sequences, or even this is both an incorporation tool, but also a search tool. So if they want to search for particular sequences, they can search by their names or identifiers, and out will come like a subtree view, which through which people can see, or the users can see where their sequences fall within the context of a much larger phylogeny. So this is the next strains auspice view, which a lot of users seem to be interested in and frequently use. So as a result of this, these tools that we have developed, we've actually enabled several applications in genomic surveillance. So the most important one of them has been to identify and name new variants in real time. In fact, Usher is the de facto standard through which all the variants get named. And till date, something that's been unprecedented for any pathogen is that more than 3,500 lineages have been named for SARS-CoV-2 till date, which means approximately 2 lineages are being named every single day. Now compare this to some other pathogen, let's say like tuberculosis. Their scientists discuss typically 7 or so lineages, L1 to L7, which have been divided based on the geography, right? So they don't have this fine-grained way of discussing what's going on in that pathogen, and that's what we'll be doing with SARS-CoV-2. So the way this has been enabled is the users who collected new sequences, like I showed you earlier, they can paste it on the Usher's web interface, or upload it on Usher's web interface, and they will see this particular view. In this example, the new sequences have been incorporated right here on this long branch. These are 3 new sequences that you see. And this example that I've shown here is actually special because this particular tree view was shown as an evidence for emergence of a new variant by Dr. Tom Peacock, as you can see in November of 2021. This variant was later designated as B11529, that has been shown here, after it went through a manual curation effort. And within a single day, this was named this particular variant, B11529. And in fact, as you can see, that some volunteer has also designated it as this variant name, but has also said that it's actually urgent, right? And in a few days, WHO called it the Omicron variant, right? So this is really an example of how we've been doing this, identifying new variants in real time. Remember that there were only 4 sequences that were deposited on the databases, and within a day or so, we've actually started tracking it very closely. So that was the pace at which we've been doing, or the larger scientific community has been contributing to the surveillance efforts. Secondly, Usher has also been a tool for tracking these variants which are in circulation. So for example, if you look at the CDC's nowcast estimates, right now you see that JN1 is starting to take off. There too, I think the way this happens is the labs actually use Usher to find the classification of that particular variant, and that data gets deposited and aggregated by the CDC, and that's how these estimates do get created. Now, people were using Pango tools earlier, which use some machine learning techniques. So we did a study jointly with the Pango group. As you can see here, this is going to be published very soon in Virus Evolution. And what we found was this machine learning techniques used to be very unstable. What that meant was from one version to the next, as the models have to be retrained, right, with new lineages and so on, with new data. And what we found was these machine learning models tended to be very unstable. What that meant was for the earlier sequences, with the newer versions, you will actually see that the lineage designations could change, which they should not in most cases. And we found that those models had like 78% consistency, whereas what we did was to integrate Usher into the pangolin tools, and then it became 97% consistent. And now Usher is the default within the pangolin tool itself. So it's basically the default tool for classification of variants in clinical sequencing efforts. But it's not just clinical sequencing where we played a role in tracking variants. It was also wastewater sequencing. So this is the UCSD study which I've highlighted with Rob Knight's group and Christian Anderson's group jointly working together for doing wastewater monitoring on our campus and in the county. Pretty famous studies, but even their tool that they developed, Freya, that actually uses Usher's mutational barcodes. So remember, we are not just giving you the phylogeny, but also the mutations annotated on that phylogeny. And that tends to be a very easily consumable information for a lot of our users because, for example, in this tool, the mutational barcodes were used to then match up with what they see in the wastewater samples and then predict the variants which are in circulation. We've developed our own tools which help us analyze transmission clusters, and we work with a lot of public health officials in this space as well. So one such tool which my collaborator Russ's group led in developing is called Cluster Tracker, which is also used by the CDPH. There, what we do is we can use the Usher's giant phylogeny, and we can predict which clusters exist in any geographical region. For example, let's say this particular cluster was found in Texas, as you can see from the sequence names, and some of those have been highlighted with these red circles. And you can ask this question, where did this cluster come from? Was it introduced from an outside state or a country? And as you can see at the root, right at the root, you see a lot of these California samples just outside. And so here from this, you can predict that this particular cluster might have been introduced into Texas from California, for example, right? And this is again, just visualizing trees and so on that helps us do this. And since this is not the only tool, actually, many outbreaks all over the world, including in low and middle income countries, have used Usher to analyze their outbreaks. We have also enabled a lot of evolutionary biology. For example, this is a famous study from Jesse Bloom's lab at UW. And what they did was to use Usher's mutational informations, essentially, to study the mutational spectrum in this virus. So again, by maintaining one single tree, which has comprehensive information of all available genomes, we enabled our users to not worry about doing their own computational analysis, but just focus on asking the right questions, right? So another such question, and I've been involved in some such studies as well. So for example, with in collaboration with Chris, who actually led this study, we showed that there are variants, for example, which replicate in the upper respiratory tract and some in the lower respiratory tract of the lungs. And there you see a different mutational signature, right? You can actually use the mutational signatures to actually kind of also guess where it must have arisen. And so the point being that maintaining this tree helps those people who, like, it relieves the people from the computational efforts, but they can just focus on asking the biological questions. And those are the sort of studies that we've enabled through this. And also there have been efforts on timing the trees. A lot of the metadata associated with SARS-CoV-2 tends to be very noisy. And so people have developed ways to time these trees on a very large scale. Chronomental is an example of that. And in fact, this also tends to be a good way of fixing the noise in the metadata. It's literally people making typing errors like 2023 instead of 2021 or vice versa and so on. And so in some cases, you might think with the metadata that Omicron arose in 2020 or something, but it's actually much later, for example. A lot of people have actually been using our tree for predicting the fitness of new strains as well. So they develop these models because it's actually big data. We actually unleash the potential of big data analysis through our mutation annotated tree. So this is an example of that. They took the 6.4 million SARS-CoV-2 genomes and they used the mutational information that Usher had released, and from there they trained their models to then use the mutational signatures of strains to predict how fit they are. And as you can see, this is actually a pretty good predictor in most cases. Here you see the Omicron variant, there's a Delta variant here, Alpha variant here, and so on. Again, this has also encouraged a lot of AI and machine learning applications. For example, people are now also trying to predict the next variant. So because, again, this is a ginormous tree with lots of data, you can use this data to train. If you can predict, for example, next mutation, or if you give a part of the phylogeny, what happens in the future in the unspecified region. So, and these models are also getting pretty good at predicting future variants. So you might be wondering that some of the previous tools had failed to scale at the pandemic level data that we generated. So what made Usher so fast? And I summarized this, and I probably won't go into much details of this, but I summarized this as four important factors. One is the choice of algorithm. In this particular application that we have, because we are generating very dense phylogenies, right? So we are sampling another SARS-CoV-2 sequence even before a few mutations arise in it. Not sampling that fast. Actually, maximum parsimony approaches work pretty well. We've shown that compared to maximum likelihood approaches. And then this is the one which I will emphasize much more in the later half of my talk, which is the use of efficient data structures, which is mutation annotated trees, right? On the tree, we annotate what mutations we infer to have occurred. And then we of course, when we are doing placement of new sequences onto the tree, we've already pre-processed all the data, right? And stored it in the form of this mat, right? So for us to incorporate new sequences becomes really easy. We don't have to really take raw sequences or multiple sequence alignment and analyze what it takes to place the sequences. And finally, we've done a lot of parallelization work, which has helped us get to the scale. And it's not like we didn't encounter scaling challenges during the pandemic. There was, in fact, we've had a lot of difficulty to keep up with the data ourselves. So the first version was probably not as good as you can see. So what I'm showing in this plot is the number of sequences in the tree, and as you can see, that has cumulative number of sequences that has grown over time. And the second plot is showing the number of new sequences which were added, and the last one is showing the amount of time that Usher took to incorporate those new sequences. And as you can see, even in late 2021, we were taking more than 24 hours to incorporate the new sequences which were added to the database that day, right? So we crossed our time limit multiple times, and so we had to then, that would typically overflow to the next day, and we had difficulty releasing the match every single day during that time. So in the middle, what we did was we actually decided to parallelize Usher with on multiple servers, and then we had another strategy to merge those back into a single tree, and that did help in the second phase in late 2021 to early 2022. But again, like we were seeing 100,000 or so sequences every single day during that part of the pandemic, so it was very hard to keep up. And that's when Cheng, one of my brilliant students who just recently graduated, he came up with really new parallelization strategies that gave us 1 to 2 orders of magnitude speed up. And since then, I think we've never hit the 24-hour limit. A lot of the techniques that he developed, Cheng developed, have been described in this particular manuscript, which came out last month in Nature Genetics. But also, if you are interested more on the whole journey, there's also a preprint that we had released earlier, which also describes some of the techniques, which I won't go into detail in this talk. So one of the questions that we've been asking is this whole Omicron example that I gave you earlier, right? That was actually led by volunteers. So they would upload their sequences on Usher and then see if this looks like a new variant that should get its own name, and then propose a name, and then some other volunteers would then curate it and then finally give it a name, right? So of course, this is a time-consuming process, and what we've been thinking recently is, can we automate this whole process, right? So can we, because we're already generating the tree, it should be possible to replicate what the human is doing. Also, we want to try to get the human involvement as low as possible in this whole process. And this is important because the lineage naming needs to continue, and it cannot be just relying on these volunteers who really have very little incentive to spend so many hours trying to both identify as well as curate lineage names. But it is very important for having a clear and concise scientific discourse. Like I pointed out earlier, when someone says B11529, we know exactly what mutational signature we are talking about, for example, right? So this should continue. So we've been thinking about, we've been working, so this is the work that has been actually led by my collaborator, Russ's group. I'm kind of involved as well, but this particular software that we have, Autolin, can identify new lineages directly from the input phylogeny, right? So there's a manuscript coming up very soon in Nature Microbiology based on this. And what we found is a lot of the effort that humans are taking can really be automated, and so we are not fully there yet, but this is an ongoing work that we've been doing. And of course, just besides naming lineages itself, there's one particular type of lineage which is really important, which is that of recombinants, which are like these hybrid strains, like Delta and Omicron recombining to form Deltacron, for example, right? And this is the way in which the virus can actually make rapid jumps in its fitness. So it's very important to track them. And they, of course, violate the normal tree structure of evolution. They create like these networks, right, that you have. And in the lineage naming nomenclature system, of course, these have like a special naming style or naming convention. Typically, the names start with the letter X. So XBB is an example, famous example of a recombinant lineage. But what we found is many recombinants are currently getting overlooked. Right, so we have developed a software called Ripples, which automates the discovery of recombinants directly from the Usher's tree. For example, this was published in Nature in 2022. And of course, it was not easy thing to do with tens of millions of sequences now. So we had to automate it, and again, 2 brilliant undergraduate students that I've had in my lab, Kyle and Cheng, have developed this tool called Rivet. And now with Rivet, what we are able to do is fully automate the discovery of recombinants. And we also have maintain a web interface through which people can curate these recombinants. So all the recombinants that we discover, and now there are thousands of those, you can actually click on it, get these SNV views, and we also tell you where the breakpoints are, which particular lineages have recombined, and so on. But the most incredible thing is we can actually do reanalysis with millions of SARS-CoV-2 sequences in just $4 of cloud computing costs. So we can literally update this every day. We are doing it currently on a weekly basis. This report was published last year in the Bioinformatics Journal. And of course, Kyle has been again leading some of the efforts in this space and doing some really cool work. So what we have found is that, and some of this work we are doing jointly with Joel Wertheim. So what we are finding is that even though Pango group is naming the recombinants, the manual discovery that they typically do is very challenging, and Rivet tends to discover far more recombinants than actually get annotated by the Pango group. So this plot is basically showing how many, the orange line is showing how many recombinants we have identified at different time points, and the green plot is showing how many are actually annotated by the Pango group. And there's a huge difference there. And there's a reason for this. The Pango group is only interested in annotating fit recombinants, right? So the recombinants which are spreading and have a lot of sequences. And so here's an example of what I just said. So we did find, or they rather found, a recombinant between these two lineages. And they found like there are 12 sequences and 6 countries where sequences got deposited. And in fact, Kyle provided them additional evidence based on Rivet that this does look like a recombinant. But this particular GitHub issue was closed because this recombinant did not grow enough in size. So they didn't even annotate it as a new recombinant. So what I mean is, if you really want to do unbiased study of recombinants, Rivet provides you like a much more comprehensive data. And what if you just look at what Pango provides, that's actually only biased towards the fit lineages. So there's a reason why I'm saying this, because it's not just Autolin. Autolin does not look at recombinants. Rivet does. So what we think is the way we will automate the variant naming pipeline would be a combination of Autolin and Rivet together. And we think that this could significantly reduce the delay in naming variants from days to even minutes. But of course, we think that some human curation will still be necessary. Okay, so I've presented all this work as just as a motivation. Now I will tell you like why we really need ultra large pangenomes for future genomic surveillance efforts. So most of you must be aware, but for those of you who are not, let me quickly summarize what pangenomes are. Pangenome is just any collection of genomic sequences which get jointly analyzed, right? Typically, they belong to the same species. And the reason why people do pangenomics primarily is to reduce the reference bias. Previously, like most of the biomedical research would happen with a single reference genome for a species, and we would do comparisons only to that. Whereas pangenomics is this idea of doing some sort of all-to-all comparisons of all the sequences that we have for that species. And the type of pangenomes that are most popular are graph pangenomes. And so the idea here is that you have these multiple sequences that for a particular species, and then you actually divide them up into the variations that you see. And this graphical representation is sort of the representation of the variation that you have in those sequences, right? So for example, in this particular region, you see the red and blue as 2 separate variations, right? Yeah, like I said, graph pangenomes are very popular, and one popular format within that is the GFA, which is a human readable format. And the way it does it is it breaks up the sequences into nodes and edges. The nodes actually correspond to the individual sequences or subsequences within those the collection, and the edges are how they are linked together. And then the paths correspond to actual haplotypes, right? So in this particular case, this path S1, S2, S3, and S5 is forming one single haplotype. So you just combine those sequences that you see at those nodes, right? And there are many extensions of this format. VG and minigraph are like variations of the GFA. Minigraph, you might have seen that in its publication that they decided to focus primarily on the structural variations. Whereas VG uses a completely different approach. They try to represent all variations. This includes single nucleotide variations, small indels, and structural variations, all of it. The problem that we've seen with this format is that these paths, if you see, right, a lot of haplotypes share the paths, like long contiguous paths. And I think Benedict, he presented last quarter in the seminar series, and he had mentioned this as well. What typically happens is that 90%, 97% or so of this format, the data in this format is consumed by storing this paths itself, and they have a lot of redundancy. So I think Benedict's group did try to fix it through this new format called GBC, which I think he mentioned very briefly in his talk last time. But I would like to say that I actually don't prefer graph pangenomes a lot, and I'll list down the reasons for those. We've tried to explore whether we should do graph pangenomes and accelerate it and try to build really large pangenomes for genomic surveillance efforts. But there are some like inherent problems with these pangenomes. I'll mention them one by one. One is that they do not store any evolutionary information, right? So they store the nodes, edges, and the variation that you see in the sequences, but there is no phylogeny, for example, no concept of phylogeny. So that has to be somehow inferred. It's not even a multiple sequence alignment, so you cannot really get an MSA out and then just pass it to a phylogenetic tool to get it. Whereas if you look at something like a mutation annotated tree that provides you the phylogeny, but also it annotates mutations on that phylogeny, right? So it's telling you not just mutations, it's telling you the evolutionary information. And because mutations lead to variation, it automatically tells you about the variation that exists in those sequences as well, right? So any sequence, for example, can be reconstructed from what you have at the root sequence, apply these mutations, and you get the haplotype at the very end, right? So it's like that storing of the paths in pangenomes. The second problem is, like what I mentioned, it has no mutational information. Only variation is stored, but how that variation came about, you don't store. So that has to be somehow inferred by evolutionary biologists, right? How that variation actually did come about. And that's, I think, a problem. Those of you who've actually created the graph pangenomes would also know that they are very hard to visualize, right? So this is one of the visualization tools that people have developed called GFA-Viz. As you can see, it's like really a mess, and I think it makes the problem harder than easier to actually view sequences this way. And this is, I'm just showing you a very small portion as compared to the whole pangenome, right? Whereas for something like a MAT, the visualization is really simple. So that you see, what you see here is what we created, like a treenome browser view for the mats that we have. So you not just have the tree also have this lineage information. So that's how they are being colored. But on the right, what you see is the genotype information. So compared to a reference, at what positions, what mutations take place. So it's a very similar to a genome browser view. So an easy way to see variations plus phylogeny. The other problem with graph genomes is that they are not easily extensible. For example, with metadata. So on our MAT, for example, we can, just like how we annotate mutations, we can also annotate the lineage names, right? So this particular branch has been annotated as lineage A, this one as B, this is B.1, and so on, right? And this is how this particular MAT has been colored, as you can see. And most graph-based pangenomes, they don't provide extensibility. VG might be an exception here again, but the reason for this is very simple. Most of these pangenome formats, the reason why they were created was to reduce the reference bias, and they had a single application in mind, which is to do read mapping, right? Read mapping and genotyping at the very end. So they really didn't focus on supporting some of the evolutionary studies that we had. I come from a computer architecture background, so it's very important for me, like, to think also about acceleration and so on. To improve the scale of approaches. And what I've found is that graph pangenomes tend to be really hard to accelerate. So here what I'm showing is how you would align, for example, a sequence to a graph. And as you can see, the dependencies tend to be very irregular. The parallelism is irregular. The memory access is irregular. But also there's dynamic dependencies. It depends on the property of the graph. And there's dynamic memory requirements as well. So for example, to store traceback path or traceback pointers here, you require more bits than you would require in some other cells, for example. So it's really hard to accelerate these techniques, whereas what we see with maths is that they are much easier. I should say that graph-based pangenomes are not the only pangenomes. There's some other work, for example, Pangraph has studied some of the limitations of approaches, and they actually end up also storing phylogenetic information in addition to the graph itself. And AGC recently was created by Henley's collaborators, and the way I think of it is it's just a fancy gzip compressor. So if you give a collection of sequences, the whole point is to store them in a compressed form. So it focuses on storage and retrieval, but not on read mapping and genotyping, which some other graph-based pangenomes focus on, and definitely not evolutionary information. So like I said, MAT is really useful data structure because it provides tree topology, it provides the root sequence, it provides the mutations and so on. And any sequence can be inferred. For example, any tip sequence can be inferred by just taking the root sequence and applying a set of mutations, and you get that sequence at the end. However, the Usher MAT, the problem with that is that it is first of all reference based, right? So the reference tends to be the root sequence. So if new sequences need to be incorporated, we first compare and identify what changes do we see compared to the reference sequence, and then we figure out the phylogeny with respect to it. So it does have some reference bias issues. It also ignores indels, right? All I showed you was substitutions, single nucleotide variations. So, but what we are seeing, especially recently with SARS-CoV-2 as well, that indels sometimes comprise the lineage defining mutations. We are seeing more of that with Omicron, for example, and a lot of our users have told us that we should try and incorporate indels into MAT as well. And finally, I think one problem with Usher's MAT was that it focuses only on the tree topology, whereas we know through the evolution, I also told you about recombination, that it cannot represent these complex mutations. For example, recombination is one of them, but horizontal gene transfer, which disrupts the vertical mode of evolution, is also not represented. So with that in mind, we have created our own pangenome data format recently, which I'm going to be presenting about today. It's unpublished work, but I think some of the results are really interesting. So just to summarize, I told you about a number of pangenome formats. All of them have focused mostly on representing genomic variation or doing some sort of full sequence encoding of the collection. Except for Pangraph, none of them store phylogenetic relationships of those sequences, and none of them store mutational information, right? So two brilliant grad students from my lab, Sumit and Harsh, have worked on creating this PanMan format, which addresses all these limitations. So you not only have mutations and phylogeny, but you also encode the sequences in a very efficient format. So that's what I mentioned here. You get for free from this format a multiple sequence alignment, a phylogeny, mutations, all in a single format now. No need to store them separately. So it's not only information rich, but as I'll show you later, it's also really compact and scalable. So how do we incorporate insertions and deletions compared to the Usher MAT that we had? So the problem really boils down to a coordinate system, and we used a multiple sequence alignments coordinate system where we treat the gaps as special characters. So the idea is very simple. If you know the multiple sequence alignment and if you know the phylogenetic tree, then you can apply some sort of like Fitch algorithm or parsimony-based algorithm or a maximum likelihood algorithm for inferring ancestral sequences. And from once you infer the ancestral sequences, you know how a child sequence has changed compared to its parents, from which you can actually annotate the mutations. So at the very end, a PanMat can store a root sequence, a single root sequence, and in each branch of the tree, it can store what mutations have occurred, just like a normal MAT. But now gaps are treated as special characters, and so you can also represent indels. Now, the question is, you have indels, but you don't have structural variation. So structural changes also take place. Large blocks move around, for example, in evolution, we know that. So there are rearrangements and so on. So it would be nice to also be able to identify homologous blocks and represent block mutations separately, right? So these are like translocations and so on. So that's also possible. We have now fairly mature whole genome aligners, which can take a collection of sequences, identify the homologous blocks. Once you've identified it, you can actually align them, just like you align nucleotides. We have ways to do that. And then if you know the phylogeny, you can then also figure out how the blocks must have mutated. And so at block level, you can create another block mutation table, which gets stored as I've shown here. But that's not enough. In evolution, you also, like I said, have complex mutations. So PanMan is a generalization of what I just described, which is PanMat, and that's to store complex mutations. So here the idea is that we not only think of a tree, but you think of a network. So let's say two nodes of a tree have recombined, right? Let's say they have recombined. Then you can just link them by network edges. That creates like a new PanMat. So now you have a collection of PanMats which are linked together by network edges, and those network edges can store information about the complex mutations. For example, a recombination where the breakpoints have occurred or where the horizontal gene transfer has occurred. So we've been working with this PanMan, and we've been now trying out various pathogens. And so what I've shown you here is the compression that we get with respect to various other pangenome formats. As you can see, compared to VG and Pangraph, for example, we get 2 orders of magnitude compression for SARS-CoV-2, but also GBC, which actually incorporates that path compression in the VG format. With respect to that also, we are basically the most compressible format that we've seen. Same is true for E. coli, HIV, RSV, all of that. No matter what we've looked at so far, we are the most compressible format, which is very interesting because we actually store more information as well, tuberculosis as well. But more importantly, we are also more scalable. What that means is as you add more sequences, we actually, the gap tends to widen between us and other formats, right? So the point that I'm trying to make is that as we get to million sequence pangenomes, we will probably be the most scalable, which is good news for us. Probably the only competition right now I see here is GBC, but again, less information there. Now, some of you might be thinking that a lot of what PanMan, the utility of it depends on what method you use for inferring ancestral sequences and annotating mutations, right? And PanMan as a format, it doesn't bother about like what you use. So we've tried both approaches. We also use parsimony, we use likelihood, and now there are good likelihood approaches for inferring ancestral sequences. So example is FastML. And what we see is because the likelihood approaches increase the number of mutations that you see on the tree, the file sizes do actually get larger, and you can see the ratio of file sizes of likelihood versus parsimony right here. In tuberculosis, we are seeing 8-fold increase with likelihood. So that's pretty high. We are looking into it, why that is, but that's not very surprising too. So Harsh has also worked on developing a utility. So PanMan is just a format, but now we want to make it usable for biologists. So for that, we've developed a set of useful utilities, just like how we did for mutation annotated trees. So if you give this utilities a PanMan, you can get a summary output, like how many nodes are there in the tree, what's the average height, what are the total number of mutations, and so on. You can extract out raw sequences, obviously, right? You can descend from the root to individual tips, apply the mutations, and you can get every single sequence. And it's pretty fast. You can also extract the variations. So this is the VCF extraction. Surprisingly, a lot of graph pangenomes don't support this. They cannot extract out VCF, even though they use VCF to generate the graph pangenome. They actually don't support VCF extraction. You can extract out subtrees and create sub panmats or panmans. You can extract out even GFA, so we are cross-compatible with VG and other tools. We can extract out the multiple sequence alignment, and we can do search and annotation, which means if you want to annotate, let's say, a new lineage, easy to do with PanMan. Okay, so that's about the format. Now I'll talk about some of the ongoing work, because the reason why we developed this was to scale to really big pangenomes. And what we've realized is that the construction process tends to be pretty complicated. It requires taking the raw sequences and then generating the alignment, identifying homologous blocks, and so on, then inferring the phylogeny, and then finally combining all of it to figure out the whole evolution, how it occurred with PanMat, right? So fairly complex, and it does take a lot of time. We are right now working in a prototype type of stage where we take small sequences and try to figure out to construct it. Right now, we don't have our own construction pipeline. Rather, we rely on PanGraph and PGGB to help generate the MSA and identify homologous blocks. But as you can see, these techniques are not very scalable. With just 20,000 sequences, it took 18 hours with PanGraph and 7 with PGGB. So the next focus that we are going to have in the lab is to accelerate this whole construction process so that we can scale to millions of sequences and pangenomes. So that's one important goal. And the hope is that with this, we will be able to unleash the true potential of big data in epidemiological and evolutionary and microbiological research. But importantly, we will also be the ones who will try to maintain it, or someone will with our tools. And that would be a one-time cost, which will serve lots of users. And so it saves users from months of computing costs, but they can just focus on asking the right questions. It would be important if a single comprehensive pangenome is maintained this way for it to have high accuracy for diverse applications, but also species. So that's another goal that we have. And we want to be able to incorporate new sequences in real time. So this is for like surveillance type applications, right? So so you can figure out the relationship of new sequences with the existing or the previous sequences, right? And as a computer engineering group, we are also very much focused on scalability issues. So we are looking into how to build algorithms which will, for example, work well on GPUs and so on. And we are actually doing GPU implementations ourselves. Okay, so as I told you that this will require at least two components. One is to accelerate phylogenetics, right? And the other is to accelerate alignment. Let me talk about how we are thinking of accelerating phylogenetics. So one very important insight that we got with our work with SARS-CoV-2 is that the phylogenies are getting very dense. just as the amount of sequencing that we do or the cost of sequencing goes down, we just sequence a lot more of any single species, and as a result, we get to be denser than sparser. What that means is we have more closer sequences that we tend to sequence, right? So with that in mind, and likelihood is obviously the preferred approach for doing phylogenetic tree reconstruction. But because of this trend that phylogenies are actually getting denser, you can actually start now applying some approximations that earlier tools did not have. They are trying to infer it accurately, but if you want to really scale, you should be applying approximations. So we've had some work in collaboration with Nick Olvin's group that developed this tool called Maple. And the idea there was to use some sort of approximation which is inspired by Asher. So it calls it parsimonious likelihood. And this tended to be orders of magnitude faster than traditional ML tools, but it was equivalent or even better results in some cases for the phylogeny that we saw. So we are thinking about these approximation algorithms that will work well for these dense phylogenies. And like I told you that the trend is as the cost of sequencing is dropping, phylogenetic density is increasing, and that's particularly helpful for pangenomics, where you're dealing with a single species. I won't talk more about get into more details in the interest of time for phylogenetics, but let me also mention that we are working on accelerating alignments. One particular strategy that we found really useful is tiling. It's something that I've been working on since my dissertation work. But recently, Sumit developed a really cool approach, which is called TALCO, that deals with tiling of alignments using convergence of traceback pointers. And so if you are working on sequence alignment problems, I think this actually you will find really useful. It works. This particular strategy works for any banded alignment and does not affect the quality of the alignment. And it does that in two phases. So it observes this property that traceback pointers tends to converge. They have weak dependencies. So the way we do tiling is, especially when you have long sequences, we divide it into two phases. In the first phase, we store the traceback pointers, and then we wait for it to converge in the second phase. So you know exactly where the traceback path would be when for longer alignments. So all longer alignments after the point after they have converged would pass through this particular path. That's the property that we use. As a result, what that means is that you can perform alignments with very little amount of memory, really long alignments with very little amount of memory. That's basically the idea. And that's very important from an acceleration point of view, particularly when you're dealing with accelerators such as GPUs and FPGAs, because they have very small amount of local memory, and they use the same global memory like the DRAM, which is very expensive to access both in terms of energy and in terms of latency compared to these SRAM memories. So if you really want to, most of these applications tend to be memory bandwidth limited. So if you really want to accelerate it and parallelize it, then you should be leveraging the reuse property of these SRAMs, right? So that's what TALCO is allowing you to do. It's allowing you to do arbitrarily long banded alignments without any compromise on the quality while using the local memory for traceback, right? That's the idea. And what we see is that it has approximately constant memory requirement no matter what the sequence length is, even in software. So this is not just a hardware approach. So for example, we see roughly 40x improvement in memory usage compared to the X-drop algorithm without any change in the quality. Compared to WFA, we see, or WFA wrap, we see roughly 60-fold improvement. I should also point out WFA group has also been working on another strategy for reducing memory requirement like by WFA. But that also increases as the sequence length increases, the memory requirement increases for it, whereas we roughly stay the same. The only reason why you're seeing a slight bump is because you need, this is in software, so you need more memory to store the actual sequences themselves, for example. So yeah, the idea is that we can apply TALCO to many dynamic programming algorithms, and this is not just limited to alignments. Dynamic programming is used in many applications in bioinformatics, as you must all be aware. It is used in multiple sequence alignment, hidden Markov models, RNA structure prediction, and so on. And TALCO is allowing us to do really long or really large dynamic programming matrices with very little amount of actual memory usage. So this particular work has been accepted to the HPCA conference, and these are the contributors. It includes 2 graduate students who had worked in my lab, and it's going to be presented in 2 months in Scotland. So yeah, this is basically nearing the end of my presentation, but we have a lot of, like I said, we have a lot of interesting work going on. So what we are thinking about in the future is to enable real-time genomic surveillance with PanMans. And now we can actually also analyze intels and other structural variations with the help of this format. And we want to apply it for diverse pathogens, RSV, monkeypox, tuberculosis, and so on and so forth. We also want to develop the support for incorporation of new sequences like what Usher did for MAT. We want to build an Usher for PanMan as well. We want to automate the lineage designation for various pathogens. So this online pangenomics is the idea of being able to incorporate new sequences onto an existing pangenome. And we are also looking forward to extending our work for some other applications in environmental and metagenomic sequences, example, wastewater. There are some students in my lab working on this particular project. And we also want to aid genotype and phenotype association studies. So for example, antimicrobial drug resistance and vaccine evasion in various pathogens. And finally, we don't want to just restrict ourselves to prokaryotic genomes, so just bacteria and viruses. And at this point, we also want to extend PanMan with our insights to eukaryotic genomes in the future. So that includes human pangenomics as well. So that's it. Thanks a lot for your attention, and I'm happy to take questions. Oh. So what Doug has asked with respect to the metadata, whether it only includes one property or you can add multiple properties, right? So whether it's just geographic location or timestamp symptoms, how is it selected? So the format is actually extensible. You can choose how to use any particular field. We just provide you a field in the format. So we use, for example, one field for storing mutations. The other field is used for storing lineage designations. You can add to a date of the sample. You can also add geographic location and so on. So that was the whole point, that users can extend with metadata the format as they please. What are the predictive use cases that Usher is currently being designed to solve? So one predictive use case is, like I mentioned, I think later in the talk, that it is used to predict what future variants are going to emerge, or which variants which have just emerged will likely dominate in the future, so which have increased fitness. Those are sort of the predictive applications. Next pangenome data set that you'd like to use for use Usher for. So yeah, we are planning to use Usher for multiple pathogens, as I showed later in the results, RSV, monkeypox, tuberculosis, and so on. So it's an ongoing work. Yeah.
What do you think are some of the largest hurdles in making PanMan applicable for eukaryotic cells?
It's a good question. So one thing is, right now, the way we've structured the format is that we are handling single chromosomes. So that's definitely going to be an issue. The other is just the scale of it, right? As we deal with larger and larger genomes, we tend to encounter like challenges which we didn't typically anticipate. So that's going to be another such problem. But it remains to be seen. We've not tried it yet on eukaryotic genomes, but all of these approaches will have to scale significantly to be able to handle genomes of those size. Yeah. Yeah, that's how we do for SARS-CoV-2 too. We've automated. We have a script, like a cron script, which every night will download sequences from a public database and just incorporate those new sequences that we find onto the MAT. So that's how the MAT do get maintained. It's, by the way, not me doing it. It's actually UC Santa Cruz doing it. Angie Heinrichs leads this effort. But yeah, that's how it's done.
Another question? Great. I guess with that, we'll wrap. Thank you all for attending. Let's thank our speaker.
Thank you.